Swayam Prabha. Digital India. Educated India.
A lecture on plasmids and so on. That is like the tools that you need to do these things. And when if you consider genetic engineering and synthetic biology as a kind of art, then you know that every painter, apart of having a sense of composition, you need to know about the colors, about the brushes, about everything. So just keep in mind that regardless of the great ideas that you may have in the future, you need to have some technical knowledge of all the tools available for making the different things. Okay, so as is the case in other days, I just want to go quickly through some of the papers that I would suggest you to read for next day. I don't know whether we'll have representatives of the 4 groups. We'll see what happens later. Okay, so I selected 4 papers. 3 of them come from the laboratory or the group of Drew Endy. That is this guy that I mentioned repeatedly here. He's the one that developed the idea of biobricks, that developed the idea of POPs, and developed other ideas that later have been quite important for different synthetic biology endeavors. So he has this interesting paper on how to measure the activity of promoters using an in vivo reference standard. And this paper is interesting because of the following. The first day, even the very first day, you remember we talked about POPs. And it was one of you that raised an important question, as well. The concept of POPs is nice, but how do you measure the POPs? And as I said, well, technically it's not very easy because at microscopic level, POPs is the transcription is stochastic, and then it's difficult and so on. So well, in the future maybe other methods will be developed, but so far direct measurement of POPs is not easy. So what others propose, instead of measuring directly the distance, the activity, and so in terms of polymerase per second, say, well, what you have to do is to propose a standard promoter. By the same token that the French Revolution said, this is a meter, and then every time that you want to measure, you have to go to the place where the meter is put, make a reproduction, go to your place and see how many times your object contains that dimension. What they thought is, well, we cannot measure POPs directly, but what we can define is a standard promoter, just an arbitrary standard promoter. And then on the basis of that, every other promoter activity can be referred to the activity in respect to this promoter. So this is what they have in this paper. And I think it's interesting, conceptually it's interesting, and also practically is quite interesting as well. So the ones that take a look to it will enjoy reading it. Now, there's this other paper coming from my own laboratory that is connected to transposon tools that I discussed in the last part of my lecture today. So in this case, what we describe is the construction and the utilities of 4 new types of transposon vectors that are useful for metabolic engineering. And as you will see in the once you have the possibility to read this paper, is that we use the vectors to insert into the business part of the vector, the cargo side of the vector, a metabolic pathway for production of bioplastics. So, and then once you have the pathway assembled, then you just shot the plasmid, the transposon, to different locations of the chromosome, and then because of this heterogeneity that you have inside a cell, then different insertions will deliver different levels and different properties for this metabolic block. So that's something that I find also quite interesting and quite educative if you want to take a look to the possibilities handed over by different genetic tools. So this is a nice paper as well. Okay, now there's this other paper that is by Drew Endy and his peers here that is about standardization of synthetic biology parts and devices. And in this paper, you will see that he proposes something that I mentioned on passing in my presentation, and is to have a super big project in which different people characterize massively the input and output functions of a large variety of genetic devices, okay? So, and he proposes some examples, not many of them, but what would be an ideally characterized part. And well, he shows some examples and discusses there's a problem here, and it's the problem of context sensitivity, as I mentioned before. So you can compare the properties of one device and the other if you do it exactly in the same context. The moment that you change the context because you use a different host, a different temperature, a different medium or whatever, then standardization starts to have problems, okay? But in any case, it's a conceptually interesting, say, story that, again, whoever reads it probably is likely to like it. And finally, this is something perhaps for more computationally oriented people here, and is the concept of synthetic biology open language, okay? That, as the name indicates, offers the community with a standard for communicating the science in synthetic biology. So that is very nice. So I want to comment briefly in what I said before, that ideally, and this is something that may happen in the future, and probably some of you will go into that, say, dynamics, is that when you think on a biology project, you always think yourself working in a laboratory with pipettes, with petri dishes, with this type of things. Well, some people say, and I don't want to take sides on that, that this is a thing of the past. So in the future, we will have the experiments encoded, and then the experiment will be actually run by a robot, okay? So maybe in the future, well, I don't know whether, I mean, in your kind of curriculum, you have to, you are taught how to program and to write a code and these things, right? Okay, okay, that's very important, and it's very likely that in the future, experimental molecular biologists will not rely so much on their own devices, but will instead depend more on a robot, on the possibilities of assembling complex experiments by coding the different actions in computer code. Okay, well, in any case, we have these four possibilities. This one I showed you before, this one here. And then at the end of the session today, we try to distribute them. I'm not sure that we have people from the four groups, but we'll give it a try. Okay, great. So that's mostly what I wanted to show you today. Now, for the presentations, there are some things that I want to tell you, because I think that it's good to take this class as an opportunity not only to learn about synthetic biology, but also to learn about other aspects of science, and what being a researcher is like. So a good part of our, say, mission is to communicate what we do, right? So, and you do an experiment, you create results or whatever, at some point you have to communicate them. The ultimate way of communicating research is by writing a paper, and you send it out, and if you are lucky, then you get a good review, and then you revise the paper, and you send it, and it's published, and then that's what we produce, right? So a carpenter produces, the chairs and so on. A shoemaker produces shoes, and we produce papers. So that's the one thing that we produce that can be measured from the outside. But this is not the only thing that we produce as scientists. We also use other channels of communication, and obviously the other big channel of communication is to give talks, okay? So I don't know whether in your curriculum you have some special seminar or some special, say, part of your, education on how to give a talk, Do you have something like that? Okay, very good. Okay, because this is something that you may think that can be learned on the fly, but no, I mean, there is a whole say, I will not say technology to give good talks that you may or may not take, because it depends also on your personality. But there are some things that you should know when you give a talk. Regardless of whether it's a journal club talk, when as we have had these days, regardless whether you go to a meeting and then you have to talk to an audience. I mean, there are some things that you have to consider when you give a talk. And please, even though the ones that talked before were not told, what I'm going to tell you now, please, this is something that you may take as a kind of take-home lesson for future endeavors, okay? Okay, so you give a talk, regardless, journal club, whatever, the moment you say, hello, good morning, whatever, my name is this, okay? Please always start saying what is your name, okay? Don't go straight into, you have a talk. Okay, this promoter here goes here. I mean, no, who are you? No, you have to say something about yourself. Okay, good morning, my name is Victor, and today I'm going to make a summary of the results or what I have read in this paper that I found very interesting. To start, and then you get started. So please do not get like an elephant into the topic. I mean, just go little by little and try to get the audience engaged in what you are saying, okay? Number two, very important, speak loudly, please. I mean, you have a microphone, so like myself, maybe it's too much, but try to speak loudly. Do not speak as well, because that's really bad. That's something that you want to avoid. So please remember that audience will appreciate that you, I mean, not shouting, but try to use a tone of voice that is high enough for people to follow. Okay. Number 3 or 4, whatever. Be careful with your body language. Okay. So body language means that the way we move, the way we do things tells others many things about ourselves that you may want or not to be told to the others. Okay. So try to move. Never ever go like this. Okay. So this is the worst that you can do in a talk. If you go like this, that means that you are terrified and that you are just looking forward for that to finish. So that's be careful. I mean, sometimes, this is a normal reaction. You go like this. It's a defensive posture. You go like this, and you feel more comfortable. I feel more comfortable now being like this, But keep in mind that the message that you deliver to the audience is different depending on your body language. Next thing, look at the audience. Look at you. Don't try to avoid eye contact. So it's super important that you engage your audience by creating some type of complicity, right? By looking around and everything. So sometimes it's difficult because and it happens to me all the time, that sometimes I'm so enthusiastic about what I'm telling you, I'm carried away. So at some point I lose track that I'm talking to you, and I think that I'm in a different planet or something like that. But in any case, when you are in a standard audience, then please try to look at the faces, and normally when you got the faces, you will also receive more attention than if you just look somewhere else. Okay? Also about the slides and about these things. So we live in a image culture. Okay, so when you explain something, in particular, you have just a short period of time, please try to explain things in a graphical form. Try to avoid excessive text. So text might be good for you to follow, a kind of a script, but for the others, it's very boring. You have a kind of a slide, all text. Sometimes, in my lecture, sometimes I have to explain, standard is this, and apart this is, it's unavoidable to have text. But in a seminar, try to avoid text as much as possible, okay? So do not use, the text is for yourself, but for the audience, the text is very boring. So please also try to make your seminars or your presentations as say, graphic as possible, okay? And the one thing that is an unforgivable sin, if you are given 15 minutes, the maximum time that you can talk is 15 minutes, period. So, and that's something that, is a kind of say, naive, inexpert individual if you overdo with the time, okay? So please, if you are given, well, you give me your story in 10 minutes. Don't be 20 minutes, because people will hate you if you do that, right? So instead of sympathizing with you, just you will get exactly the other negative feedback, okay? So, well, you know, that has happened to me. And I remember with horror, the first time I gave a public presentation, I was given 10 minutes. And then, you know, when I thought I was starting to end the introduction, the chairman told me that the time is out. And he kicked me out, like literally, you know. So that means that, you know, you have to learn how to control the time. But I say, well, but my story is very complex. And then how can I tell this story in 10 minutes? Okay, there are ways, you know. If you watch TV, sometimes we get these advertisements, and an advertisement may last 15 seconds. And you can have a complete story in 15 seconds, okay? So obviously, saying complex things in shorter time is more difficult than having more time to tell stories. But you know, you have to really stick to the time. That's super important. And well, this is a kind of friendly audience, and you know, if you go over time a little bit, it's not a big deal. Even though, for the sake of training, it's good that we try to stick to the time. But if you are in a professional setting and you mismanage the time that you are allotted to speak, that will give you a very bad impression. Okay. So please, you are given 10 minutes, you have 10 minutes, not 1 second more. You have 5 minutes, 5 minutes, you have 2 minutes, you have 2 minutes. In the last few years, I don't know if you have it here, one thing that is becoming very popular in scientific meetings is the so-called the poster flash presentations. Did you ever hear about that? Okay. So one way of scientific communication is to have a poster. You know, you have a session, poster, my research, here and that. And then, you know, sometimes going through the posters is a bit complicated. The place is crowded. And sometimes, you know, you cannot really go in detail to everything. So in many cases, in many meetings, there's a special session that is called flash poster presentations. Is that every poster presenter is given 2 minutes to tell in 2 minutes the content of the poster, okay? And that's something that is incredibly successful because, you know, in a short period of time, you have a kind of glimpse, you have an overview of what each of the poster presenters are doing at any given time. And that's something that's super useful. And you have 2 minutes, and there's a clock with lights, you know, that the moment that you overdo, it starts like flashing and everything, and you get ridiculed in front of everyone if you overdo with the time, okay? So I think that, you know, I see that this is a kind of training setup, and it's not a big deal if we overdo a little bit, but please, for your professional life, keep an eye on the time, okay? And also, you know, one thing that I would finally tell you on how to make a presentation of this sort is that try to enjoy what you are doing, okay? Do not be terrified. Do not feel frightened. It's just like, you know, I mean, you are being observed, and that's the moment that you have to really deliver your message to your colleagues or to an audience, okay? So whether your English is very good, my English, as you can see, is not perfect at all. I mean, don't feel frightened, don't feel intimidated. Just the contrary, try to enjoy what you are communicating to the others. That this is the minutes that you have to communicate a given piece of science is the one moment that you have to really show yourself and your capabilities to the others. Okay, so let's try to learn that. Okay, so that's something that maybe at the end of as I have now most of your presentations, then maybe at the end I will make a little appraisal of how your presentations have gone, and then you can learn out of it, okay? And also, please consider all these like constructive criticism, okay? So if you are not told that this could be done better, you will never change, and you think that you're doing something wonderfully, and it may be not, right? Let's take this criticism and these advices in a constructive fashion, okay? Okay, so after all this little speech on how to make a good presentation, let's continue with the presentations. And we have to start with you, right? Because yesterday...
Actually, I read up a bit on the questions I found answers.
Okay, so but you left us with the last part of your presentation untold, right?
No, I completed the presentation.
Okay, If you consider that, I thought that you wanted to elaborate a little about the last part of your presentation, but if you consider that everything was delivered, so be it, no problem.
No, I'm pretty sure I completed all the contents of the slides.
Okay, perfect, very good. Okay, so then we have you, right? Okay, please check with our technician whether you can use his a computer or whether you have to make a change, okay?
So, good evening, everyone. I'm Nate Maheshwari, and I will be presenting my review on the paper on digitalizing heterologous gene expression in a gram-negative bacteria by using portable on and off module. So, uh, the the digitalizer module, basically what it does in principle is it will try and generate a digital sequence, which means that once we turn the inducer on, once we induce using the inducer, the the gene expression should suddenly shoot up, and once we take the induction off, it should become zero. The main rationale behind why this method has been employed is because the pre-existing methods use an arbitrary threshold to decide what the off state is. This method is able to get a very quick and effective off state because it uses an sRNA regulated promoter sequence. This sRNA regulation works because the sRNA is able to block translation of a repressor sequence in that plasmid, and that repressor is then translationally coupled to our gene of interest. So here, this is the a broad scheme design of the entire system. It contains two modules. There's an inducible promoter module and a digitalizer module. The inducible promoter, as the name suggests, works on the basis of an inducer that is able to induce the transcription of a transcription factor that will further cause the transcription of our digitalizer module. Okay. Right. Moving on. So there are three main parts of the system. There's an inducible promoter that will have a given level of basal expression, which we will call the off state, and it will have a strong promoter for the sRNA. But this promoter will be repressible by a repressor that will be transcripted, as we saw in the previous design by this repressor sequence over here. Moving on. The transcriptional repressor that will be induced, that will be expressed through the inducible promoter sequence, will be translationally inhibited by the sRNA, however. So the way it will work effectively will be that if the sRNA is being produced, it will stop the translation of the repressor sequence and hence also the gene of interest, which is what we will call the off state. So that is what the last point means to convey, that it is translationally coupled to the repressor gene. The translational coupling is done through various methods depending on the example. So yeah, this is a summary of how the digitalizer module will work. Could I use the whiteboard? Okay. So effectively what we have is there will be an inducible module over here and a digitalizer module over here. Now let's consider a natural off state. What that would mean is that you have a high amount of this sRNA that is there inside the cell, and what that sRNA does is it stops the translation of the entire repressor and the gene of interest. An interesting thing to note here is that this repressor is what, when the repressor is being produced, it targets the production of the sRNA altogether. So the repressor represses the sRNA production, and the sRNA in turn can repress the production of the repressor and the gene of interest. Now, consider the state where we induce, where we have our inducer that goes in, and it will start the translation of a transcription factor over here. The transcription factor, when activated, will start the transcription and the translation effectively of the repressor. And as the repressor is being produced, it will start inhibiting the production of the sRNA, which means the translation will be allowed now. Further, the repressor is translationally coupled to the GOI. So if the repressor is being translated, it means that the GOI will also be translated and hence expressed. So that is what we call the on state. Okay, and the minute we take the inducer off, the repressor is no longer produced, so the sRNA will start getting produced. And as I said, the sRNA can completely block the translation, which means it will be an immediate off situation for our gene of interest. Okay. Moving on. So, computer simulations were run for this model, and it was seen that at the strongest levels of repression, which is shown by the red lines over here and here, we see that the digital behavior is observed sharply when the repression is at its highest. So here the MS represents the number of sRNA molecules, and the MR is the mRNA that is produced due to the transcription of, of the repressor and gene of interest. Clearly, the MS is inversely proportional to the MR, and over here we see that this P1, which is the strength of the promoter, also which is the same as the amount of transcription factor molecules produced. Beyond a certain point, there's a sudden jump in the amount of MR produced, which also goes to show that there's a sharp off and on that is observed. Moving on. So we now have an example that was constructed. So here the Xylus was used as a transcription factor for the LACI repressor produced, and the LACI repressor is translationally coupled to a GFP to measure the quantitatively measure the repression. Further, we use a MIXI sRNA, which includes an HFQ chaperone. However, the MIXI was redesigned so that it will, when it's produced, it will stop the translation of the LACI repressor and hence also the MSF GFP. Moving on. So once we simulate this example, we see that the GFP, the fluorescence of the GFP, clearly you can see that once we induced, this line here represents the concentration at which the induction occurs. So below that concentration, the fluorescence has a certain value, and beyond it, it has another value. Excuse me. There's clearly no overlap, which again goes to show that there is a sharp on and off functionality that exists. Further, to demonstrate the high accuracy of this method, two more experiments were conducted using the NIS protease, which is sensitive. So a TPI protein which is sensitive to the NIA protease, which when on, the NIA cleaves the TPI, and this can be observed in an assay where we can clearly see that the TPI was cleaved when it was induced. The second test that was done was we substituted the MSF GFP by a collicine E3 gene. Now we must note that even one molecule of the prolisine E3 of prolisine is fatal to the cell, which means that if you induce the production of the prolisine, it should immediately kill the cells. This is the observations from the prolisine experiment. So here we can see that beyond a certain concentration, the cells have clearly been wiped off, and below a certain concentration, they're still there, very much there. And here we can see a graph of the cell growth. Beyond 16 hours, the cell mutates to become immune to the prolisine E3 gene, prolisine. But below that, we can see that the cell clearly dies once induced, otherwise it proliferates. So, to do a review of this paper, the points that I liked about this paper was that it acknowledges that other than the digitalizer module proposed here, there are other methods that do the same thing, but it still says why the digitalizer is in many ways better or more accurate. Then it used multiple methods to validate the on and off response. As you saw, it used the GFP, then it used the NIATPIA method, and then finally the policy method. One of the points I kind of did not understand was it mentions that high repressor and low sRNA also keeps the system off, but it does not elaborate on how it does that. That's it. Thank you.
Thank you. Well, thank you. You made a very good job. Thank you. So, what do you think of the paper? Forget the fact that I'm one of the authors, okay?
So, I mean, I like the concept, but when I was reading more about the paper and what it does, I also found that there are other methods to do this exact same thing. Not the same thing, not the exact same thing, but a similar approach. The similar result is obtained. So what I kind of wanted to know was what exactly was the motivation to come up with this method and not use the exact existing methods behind this paper, behind this method.
Okay, very good.
Any questions?
Questions, comments?
Thank you.
Okay, well, you know what? So obviously this digitalizer comes from my laboratory, and we have tried to use it in various contexts. And also I have to say that not in every case we have a completely digital response. Okay, so for some reason taking transcription to zero is very difficult, because even if you play with the promoters, then there are occasionally transient promoters or cryptic promoters that you don't detect and can still produce some level activity. So that's something that sometimes kills your it's one of these emerging properties that we discussed before. You know, this digitalizer is 2 + 2 should be 4, but sometimes you have this emerging property that will not fit exactly the model. Okay, so that's something that is quite good. Okay, so yes. Yeah, well, this is exactly what is being done. Okay, so as getting digital responses is so difficult, so what people active in the area do is that they establish thresholds. So if you go beyond that, you are on. If you are below that, is off. And in one of the remaining classes, I will tell you about this circuit design. And this is indeed one of the criteria, because not every promoter can be digitalized 1 to 0. I mean, that would be impossible. However, you can make the abstraction that you take a promoter and then you say, well, I put a threshold, an arbitrary threshold, and then I consider that you are on if you are below, you are beyond the threshold, and you are 0 or you are off if you are under that. But you know, the reason why you cannot have transcription 0 completely is because the intracellular medium of the cell is stochastic. Okay, so you may have a repressor, but the repressor is not sitting there all the time, and it's going in and out. And then even you have strong affinity, a strong affinity does not mean that the protein is there forever. I mean, you have a kind of affinity constant that reflects the number of times that the promoter hits the plate. So it's kind of unavoidable that in a stochastic universe, that is what you have inside a cell, at some point, some transcripts will be produced. So it's nearly impossible to avoid altogether. But for practical reasons, people do exactly as you say. Okay, so no more questions. Then I received today at least 2 presentations. Yes, please, well, each of the 2, you will both show up, yeah. But you have to load your presentation somewhere or use your own computer because, you know, I received your presentation, but they don't let me use my computer. So you have to either use yours or load your presentation in the computer lab.